So good morning again. For those of you who are in the demyelinating CMT session, repeat a few things. So it's really wonderful to be able to talk to you. Really, we're going to delve a little bit more into gene therapy. Maybe I'll switch to this mic if that's okay. Can you hear me okay? All right. So my name is John Sperrin. I'm chair of the Scientific Advisory Board for the CMTA. I have a lab at the University of Wisconsin-Madison where we work on different aspects of CMT. And it's really my privilege to not only provide some overview, but also really introduce Zach Nevin, who's the next speaker, who's part of a really impressive team at UCSF, who's developing special types of sort of genetic therapy for CMT. So again, I'm going to start off with just a reminder of the fact that when we talk about gene therapy, we're talking about trying to fix two different kinds of cell types. So for individuals with demyelinating CMT, we've talked about this, that the Schwann cells that line the nerve and create myelin are the cells that we need to try to repair. And so in this case, when we're talking about gene therapy, we're trying to deliver something to Schwann cells. However, when we're talking about axonal types of CMT, generally we're trying to deliver that therapy to motor neurons. So motor neurons have their cell bodies in the spinal cord, and they send these very long projections out to all your muscles, out all the way out to your big toe. And so these are two different cell types that we have to think about in terms of delivering these genetic therapies. So let me give you one version of gene therapy, which we sometimes refer to as gene replacement therapy. In this case, if the function of a gene is deficient, we like to deliver the correct version of that gene. And we need to use vectors. These are just a way of delivering to the correct cell type. And the most popular type of vector right now is AAV, which is a type of virus. And for those of you who were in the demyelinating CMT session, Rachel Saltzman gave a really spectacular display of how this has worked well in models of CMT1A. And essentially, we're just using that virus as a temporary delivery vector to get that correct version of the gene into the cell and restore the function of that gene so that we can essentially try to cure that neuropathy. So this was really patterned on earlier success in a disease known as spinal muscular atrophy. This has been a tremendous success story for the field with an FDA-approved therapy. There are different types of that therapy, but AAV gene replacement therapy has been very spectacular in really treating a devastating disease that affects the same cell types that are affected in CMT as well. Now, we always wanted to sort of follow this template, and we're really actively working towards that. We have actually already had clinical trials for AAV in CMT, and this was actually in a type of CMT known as GAN, GAN, giant axonal neuropathy. This was a study conducted at NIH. So those are things that have already occurred. We've been partnering and also promoting other efforts for axonal forms of CMT. This is, again, for motor neuron delivery, and these are in types of CMT classified as CMT4J, CMT4A, and CMT2E. And these are in various stages of trying to develop these therapies and showing that they're successful in animal models, and then hopefully then developing the necessary data to try to bring those to clinical trials. We did already talk about this in the demyelinating CMT session, so I'm not going to mention the demyelinating CMT subtypes in this slide. But this is really a template for this certain type of genetic therapy, which has proved successful in SMA, so this is something we can hopefully try to repeat for CMT. Now, for demyelinating CMT, just again to remind you, the problem there is really the excess amount of PMP22, and the fact that we're able to reduce that. This has been shown to be successful. in various ways in animal models of CMT. And in fact, one of the major success stories is a demonstration that this works in animal models. This was initially led by Dr. Cleopa, who's a neurologist in Cyprus, in collaboration with Scott Harper at Nationwide Children's Hospital. And if you missed the previous session, Rachel Salzman from Armitage has really shown some spectacular data showing that this looks really feasible to bring to clinical trials. So we're very excited about this approach, and so CMT1A is certainly in the crosshairs for that. So the other thing I wanted to just mention is all the other gene therapy efforts for demyelinating forms of CMT. And again, I want to highlight the work of Dr. Cleopa, who's really been a superstar in this field. He's a member of our scientific advisory board, and he's really led the way not only in CMT1A, but also CMT4C, which is a demyelinating form of CMT, CMT1X, and there's been beginnings of a partnership with Sarepta for that. We've also funded some efforts in CMT 4B1 and 2. This is a project that was recently approved by the SMA. And in all these efforts, we've really been guided not only by Dr. Cleopa on our scientific advisory board, but we've also recruited some really amazing leaders in the gene therapy field to our scientific advisory board. This includes Scott Harper at Nationwide Children's, and then Stephen Gray at UT Southwestern. And so we're really very well served by having really some superstars in these fields to help guide our efforts. So another category of what I call a genetic therapy is what we refer to as ASO or RNAi. And again, for CMT1A, as I told you, the excess of PNP22 is a problem, and AAV gene therapy is one way to approach that. But this other technology is referred to as ASO, antisense oligonucleotides, or ASOs, or RNA interference. And these are just really small pieces of RNA or DNA, which we can deliver down to sort of tamp down the level of PNP22. And this was actually a really important success story that back in 2018, the paper was published from a collaboration of the CMTA with Ionis, which showed really dramatic improvement in rodent models, be able to reduce PNP22 and improve the function of those animals. So I'm just trying to put this in perspective in terms of all the types of CMT that we're working with, CMT1A being the largest proportion of the CMT population. We really have worked on a very multifaceted effort to try to test individual genetic therapies. One of those is sort of CRISPR-Cas9, and I'm going to leave that to the capable hands of Zach, who will talk about that next. But we're very much interested in ASO and RNAi technology, and then obviously the AAV approach that Armitage has been developing has shown a lot of promise, and we're very excited about that. We've also, as I mentioned in the previous session, really tried to work on new delivery modes. We think we have some delivery modes that work, but we have to deliver these gene therapies to the correct cell type. And we've also funded another project from the lab of Dr. Cleopa, in this case led by Alexia Kajiava at Cyprus, to try to develop a new way of delivering these genetic therapies. I want to delve a little bit more into the ASO RNAi approach, because what's really sort of caught many of our attention is an approach that was originally developed by a company known as DTX Pharma. But then this was acquired by Novartis, and this was really a major landmark in our CMT studies, where major pharma really adopted an approach to try to attack CMT1A. And the thing about this was not only the size of Novartis and their ability to bring things to clinical trials, but also this was an effort that's led by a colleague of many of ours named Dr. Robert Balow. He was a neurologist at WashU and then Cedars-Sinai, and he became global head of neuroscience at Novartis. He had worked in CMT and ALS spaces for many years. And so since he knew CMT very well, he was well positioned to try to bring Novartis and all their resources to bear to attack CMT1A. He was actually a previous member of our scientific advisory board, but he had to give that up as a conflict when he joined Novartis. But he remains actively engaged in promoting this initiative by Novartis, and we're very excited about that. I want to give you a little bit of history about this because when we think about how things evolve over time, there are many steps that have to occur along the way. And I think it's instructive for us to really think about how this initiative really developed. Again, the inspiration for many of this was development of ASO therapies for SMA. And this gave us a template to follow. We were able to show the first steps in a collaboration with Ionis, and this was published back in 2018. Now, at that time, we actually were confronted by two major challenges that we had to really confront. One of which was we weren't ready at that time to really initiate clinical trials for CMT1A. We didn't really have sophisticated outcome measures or other biomarkers that we could use to support efficient clinical trial design. And so in the intervening years, even though Ionis never finalized their effort to try to bring ASOs to clinic, we were able to use that interaction to really refine and develop better outcome measures for better clinical trial design. And then one of the things that was a challenge too was that we needed to have more efficient delivery to nerve. And this was a challenge that was attempted to be addressed earlier on, but the success that we saw in this paper really inspired a number of other companies to jump into this field. And I can tell you about some cases where we've been able to partner with companies like DTX Pharma and Novartis to try to accelerate their development. But there are a number of other companies in this space that have done preclinical testing with us. I can't disclose the names of all of those because some of those are still confidential. But between Novartis and other companies that are in this space, I'm really confident that we'll be having clinical trials for CMT1A for this sort of technology. And so this was a road where we realized some challenges and hurdles that we had to overcome. But I think we're at a very important place right now where a lot of these challenges have largely been addressed. And I feel like there's relatively few barriers to get to clinical trials in CMT1A. So the other aspects I just wanted to mention is the fact, again, that delivery is a really important aspect of all these gene therapies. So we've been trying to, develop new modes of trying to deliver ASOs or RNAi or CRISPR-Cas9 or other things. And if we are able to solve this delivery issue for CMT1A, this will really kind of benefit CMT1X, CMT1B, all these other demyelinating CMT forms. And so this has really been another major effort for us, is not only to try to, have a fix that works, but also can be efficiently delivered to the correct cells as well. So overall, I just want to kind of reinforce that these efforts on the CMTA side started, 5 or 6 years ago now. And in the meantime, we have a relatively all-encompassing effort on many types of CMT. This pie on the right side of the slide sort of represents the proportion of all the CMT subtypes. And if you look at this line that goes around there, we've actually are working in about 2/3 of the types of CMT, either supporting efforts of academic groups or interacting with companies to try to develop genetic therapies for all these different types of CMT. And this reflects a lot of different technologies, not only AAV gene therapy, but other types of technologies like CRISPR-Cas9 as well. So I think that's my last slide. I think we'll have some questions later, but it's right now my pleasure to really introduce an expert in the field of gene editing. Zach Nevin, again, is a member of a really fantastic team at UCSF, who's really sort of spearheading the application of CRISPR-Cas9 to CMT genetic therapies. And he'll give us a really insider's view of how that technology works and the promise that it holds for the future. So, Zach?
Hi, everybody. Yeah, I'll use this. Tend to move around a little bit, so this might be better. So thank you for having me here today. I am a postdoctoral scientist in the lab of Bruce Conklin. That's the wrong way. In the lab of Bruce Conklin at the Gladstone Institutes in San Francisco. We're very closely affiliated with UCSF. And for the past 9 or 10 years now, we've been thinking about how do we combine all of the new technologies that have come out in recent times in order to actually start developing therapies for, in our case, we're focused on neurogenetic diseases, including the Charcot-Marie-Tooths. And so working with groups like the CMTA, we've been put in contact with collaborators who are really experts in the neurogenetics of these diseases. And our particular start into this space was coming from stem cells and using actually patient-derived stem cells to then generate neurons in the lab, in dishes, so that we can understand the basic biology of these diseases and then start to develop therapies to test in the specific models of different genes, different mutations. And more recently, with the development of CRISPR technologies, gene editing technologies, we've actually been able to start coming to the fruition of these projects and actually disrupting mutations and figuring out ways that we could potentially treat at the DNA level permanently some of these diseases. And then lastly, even more recently, you may have heard that the COVID vaccine was developed using something called a lipid nanoparticle as a way of delivering genetic cargoes to the cells as a delivery system for gene therapies, or in that case, vaccines. And we've also been thinking about, well, how can we take advantage of this to get the CRISPR therapeutics to the place where it needs to be, to have an effect in the cells, in the neurons or the Schwann cells. And so, like I said, we've benefited from collaborators who are experts in the neurosciences, through the CMTA and the California Institute for Regenerative Medicine on the stem cell side, and then also a number of collaborators who are recognized throughout in the Bay Area at large, so at Berkeley, at Stanford, and even the tech that's so prevalent in the Bay Area. And so just a quick overview, you've probably heard at this point of CRISPR, but what does that actually mean? So there's really 2 key components to the CRISPR technology. The first is the Cas enzyme itself, and that's demonstrated in yellow here, and that's a protein, an enzyme that's able to interact with DNA and break it. And in order to control where those breaks occur, the second component is the guide RNA, highlighted here in green. And that's a sequence that has this little loop structure that interacts with the protein, the Cas, and then a sequence which we can design that indicates where along the DNA the Cas is supposed to interact, is supposed to break. And so at the end of the day, if we can put a guide RNA that we have engineered with the Cas protein, it causes a double-strand break, shown here. And this is really powerful for disrupting genes. The cells don't like double-strand breaks. You could imagine in your DNA, if you have a double-strand break just sitting there, it's not good. It's not how it's supposed to be. And so we're taking advantage of this in 2 different ways. In the first, using a single guide RNA targeting Cas9 to cause a single break in the genome, we can actually use this to disrupt expression of a gene. For instance, FMN2, NEFL, PMP22. And the way that works is that after double-strand break has occurred, the cell doesn't want that to be there. It starts to repair that break. It sticks the DNA back together, but that's a very imperfect process. And oftentimes the cell will result in either a removal of a little bit of DNA or an insertion of a little bit of DNA, which ultimately disrupts the gene. And we call those indels, shown up here on the top. The other way that we can use this is actually to use 2 guide RNAs, cut 2 places in the genome surrounding a gene. And when you have those double breaks, then you can actually remove the entire intervening sequence, which could have our gene of interest, it could have a mutation. But through these 2 different approaches, they both ultimately inactivate whatever we're targeting in the genome. And so we've been applying this predominantly to dominant disease. So that's where you have, for most of the genes in your body, you have 2 copies. And with dominant disease, one of those copies has a mutation. And if we were able to remove that mutated copy, then the wild-type copy, the normal, healthy copy, could potentially reassert itself and restore health to the cells. And so this is where CRISPR really comes into play, targeting the one mutant copy to allow the healthy copy to remain behind. And we're using a system which we call a stoplight assay to really engineer and determine how effective this process can be. So in this system, we take the two endogenous copies of a gene, the two copies that are in your genome. We tag one of them with a red protein that we can visualize and the other one with a green protein. So then looking at just single changes between these two copies, whether it's the mutation, whether it's something benign but differing between these two copies, we can design guide RNA targeting, in this case, either the healthy gene, the C, or the mutant gene, the A, and ask how effectively does that actually get rid of the mutant gene? And also importantly, is there any situation where, for instance, we're targeting the C, but we accidentally hit the A? And that would be the risk or the off-target effects of these CRISPR approaches. And so what you can see here, starting on the left, is this assay just in wild types, it's just in unedited cells. So you can see that there is yellow in the center of the cell. That's where both of the genes are expressed. And then a little bit of green in the outside periphery of the cells. That's because in this particular mutation, it causes the protein to be mislocalized outside of the center of the cell. And then moving from left to right, if we target the red allele, where the red arrows are, you can see that the yellow has disappeared from the center of the cell. That means we've knocked out the red. When you look at red and green together, it makes yellow in this study. And then in the third panel, if we actually go after the mutation, the mutant allele, you can see that there are many cells which are now red only. We've gotten rid of the mislocalized protein around the outside of the cells. And then if we put them together, we can see any combination of these. And that's just a test to see kind of how they interact together. So what does this actually look like when we apply it to CMT mutations? So here in the top panel, we have iPSC, stem cell derived neurons. On the left in the A panel, these are wild type, meaning that they are from a healthy control cell line. The patient didn't have, or the donor of these cells didn't have a mutation. And so you can see outlined in blue the body of the cell. And then you can see these lines in green, which are the NEFL protein in the axons of these neurons. Now, when we look at an N98S mutation in NEFL, so this is affiliated with CMT2E, what you can notice is that in the body of the cell, outlined in white in panel B there on the top right, there's a lot more of the NEFL protein stuck in the center of the cell. It's not making it out to the axons. But when we treat these cells with CRISPR, actually targeting this mutation, in the bottom, in the center panel, again, you can see what these cells look like pre-treatment. And on the right, you can see that treating them with CRISPR actually gets rid of the mutant copy of the gene, and we're able to restore the normal distribution of NEFL out into the axons. And so this was looking at one CRISPR targeting one mutation. But as you know, there are many different mutations, even in a single gene affiliated with CMT. And so the big question we've been approaching in the lab is how can we develop a therapy that might be able to treat more than a single mutation? And so this is a very busy slide, but just to orient you quickly, all these little colored tick marks are indications of where in the genome there are differences. And each of these rows of tick marks are an individual patient's genome. And in this case, we're looking at the gene MFM2 and asking the question, where are there differences in the genome that we could actually potentially target with CRISPR? And the way to look at these is the light blue and the gray are anywhere in that particular patient's genome where the two copies of MFN2 are identical. So we wouldn't be interested in trying to target those because we would hit both the healthy and the disease gene. And that would be not great in the end of the day. But any of the dark blue tick marks here, that means that there's a single base, a single nucleotide, a single DNA change between the two copies of the gene. And that is something that we can now potentially target. And so if you look kind of down any single column of these tick marks, you can see some places where 50% potentially of patients have these dark blue tick marks. And so if we design a CRISPR targeting that particular location and know whether the A or the G allele is linked to the mutation that that patient has, we could actually develop one or two guide RNAs for CRISPR therapies that would then treat anyone with these other benign variants. So I hope that makes sense. Happy to talk about that more. But this is a way that we are designing CRISPR to treat many different mutations by targeting just one or two sites in a gene. And using the similar approach, but now looking outside of the gene, we've been working with members of the CMTA, Maurizio and John, to actually try and excise either MPZ. We're doing this study in mouse models, which are have these two changes around the gene, or in primary Schwann cells from human donors looking at PMP22. And in both of these cases, we're asking the question, can we use benign variants around a gene to take out the mutant, inactivate it, and therefore restore health to the gene? So I'll move through very quickly, 1 minute. So the other question that I told you we were asking is, how do we actually get these reagents into cells? So we've been using lipid nanoparticles. And what I'm going to show in the next two slides are just two particles. And there are hundreds of different particles out there that we're now starting to screen for their efficacy. And so for this first particle, when we injected it by lumbar puncture, or intrathecal in this case, into a mouse, you can see this green, the cargo that we're delivering. It's kind of on the outside of the spinal cord, which is outlined in all of this blue color here. So for treating neurons, that wouldn't be ideal because we're only hitting the superficial level of the spinal cord. But if we do an injection of the same particle directly into the tissue of the spinal cord, then we see very robust, very wide delivery of our marker here throughout many different cell types in the spinal cord, and beginning to seep into the projections of the neurons, the axons, as they begin to exit the spinal cord. So this is just an N of 1 example of an LMP, but we're starting to screen many, many different formulations to find the best ones to treat either neuronal or Schwann formations of CMT. And so lastly, just to kind of give you a perspective on what is going on with CRISPR more broadly. So the very first CRISPR therapy in humans in the US was approved last December, and that was actually for blood disorders. But as you can see, there are many others in the pipeline advancing through phase 2 and phase 3 trials, and these numbers will just continue to stack up. What you don't see on here yet are neuromuscular disorders, but as we've been hearing, those are very much coming along the way in CRISPR as well. And then lastly, one of the important things for not only CRISPR, but all of these gene therapies are ways that government and regulatory agencies are actually trying to improve the process for developing these genetic therapies. In the European Union, for ASO technology, which you've heard about, they've approved what's called the hospital exemption, where you can actually develop a ASO targeting a single patient for use in that single patient so long as they reside in the hospital for the entire duration. Something like that may be under consideration in the U.S. in the not-too-distant future. But the FDA here in the U.S. has also developed what's called designation for platform technology. And that means that when you have an LMP and you have Cas9 and you have a delivery method that are all going to be identical no matter what disease you're treating, and you're only changing the guide RNA, for instance, to change which target in the genome the CRISPR is actually affecting, then that would accelerate the approval process for that. You might not necessarily have to go through the whole phase in the same depth. It's just the one piece that as soon as you can de-risk it and show that it's safe, there may be a fast track into the clinic. And so we're very excited about all of these developments and the way that this is going to actually accelerate and increase that list I showed you of CRISPR technologies actually getting to patients. So with that, just to thank the people in the lab, as well as all of our collaborators, and I think we're going to be taking questions now. Thank you.
So I have a question about the CRISPR technology and who are the prime kind of candidates for it. Is the goal to get it in utero as early as possible? Is the goal to get it in patients that already have the disorder? And if it is in patients already with the disorder, what does that look like? Are new proteins then? They don't have the mutation? Or then does your body slowly start creating healthy ones and replacing the unhealthy ones? Just love to learn a little bit more about that.
Sure. So I think with all of these gene therapy technologies, there's a general sense of the earlier the better, and trying to stave off the disease. That's not to say it's not going to have an effect, beneficial effect in either halting progression or potentially even reversing disease if it's applied later after symptoms have started. With CRISPR in particular, I think one of the key things to distinguish is the protein that we're trying to get rid of, the mutant protein, and then the cell that protein is affecting. And so with the CRISPR, we're getting rid of a protein that's having a negative effect. And it really is going to depend on whether the cell is still present, how healthy the cell was before we were able to intervene, and whether the cell can then reassert itself in the system. So I think with Schwann cell disease and neuron disease, even though the disease is progressing, a lot of times the cells are still there. They might be a little tired. They might be not really behaving the way they're supposed to, but they're present. And so there is definitely a chance that removing the mutant protein will allow the cell to wake up and begin acting a little more normally. But that's going to be a disease-by-disease, case-by-case basis.
Maybe I'll just add that in spinal muscular atrophy, the first trials were actually in kids because they were really severely affected. I think in CMT, it may be that many of these therapies may be tested initially actually in adult populations. There's certainly a higher bar to do pediatric clinical trials because of the risks associated with that. So it'll vary from disease to disease, but I anticipate that some of the earlier clinical trials might be happening in adults rather than kids at this point.
Thank you. Any more questions?
It's a two-part question. The first question is just for clarification. When you say you're focused on dominant diseases, I'm assuming you're referring to autosomal dominant. And my question is, why are you focused on dominant and not recessive? Is it because it's easier to just clear off one gene than two? So that's one part of the question. Then I'll ask you a second.
Sure. So with gene delivery, like John was talking about in the beginning, that's putting in a healthy copy of a gene. And so with recessive disease, where you have two mutations, typically those mutations are inactivating the protein. So going in with CRISPR and removing a protein that's already inactive isn't expected to have a benefit. So with dominant disease, you have one healthy and one disease. And if you get rid of the disease, it allows the healthy copy to reassert itself.
Okay, that makes sense. The second part of the question is, with the evolution of artificial intelligence, is the medical community looking into leveraging that technology in order to accelerate data analysis and however else it may help?
Absolutely. Not only on the clinical side and trying to understand when there are interventions or looking at tissue samples and trying to more discreetly define a disease, but also in the therapy development side. That one slide I showed you with all the blue and gray tick marks, if we were to go through by hand and try to design a CRISPR against every single one of those, that would be the rest of my career. But we are developing pipelines to actually do that at scale, and we're trying to develop a screening method in the lab to test thousands of different CRISPRs head-to-head in a single experiment to try and figure out actually which one's the best and not go piecemeal, step-by-step, testing one after another.
It's a great question about AI. In your registration packs that's on your desk in front of you, there's a copy of the CMTA report, and the headline cover article says, "Can AI cure CMT?" And it's that one. So if you turn to that article, written by a really lovely lady, it goes through all the different ways that AI could be used to help in the development of treatments for CMT. And one of the examples we give is a CRISPR example. So enjoy reading that on your journey home.
Not to steal Catherine's thunder, but, I think it is a profound influence. It's hard to throw a rock on a university campus without hitting someone that's working in the AI space. And essentially, this is being used not only in design of these sorts of strategies, it's also being used in outcome measures like analysis of the MRI studies and analysis of other outcome measures to try to improve that as well. So I think Catherine's article is probably a good place to start it, to figure out how AI is really transforming a lot of different activities in our space.
Thanks, John. I do mention those two applications in my article, so you just spoiled it. Were there any questions on Zoom? Hi, can we get a microphone to Sarah Cader and we'll go to a question there. We've got 2 minutes left.
All right. One is, does this technology permanently change a person's genes, or would it last for a certain period of time before needing to do it again?
Maybe I'll start and Zach can finish. So it depends on the type of therapy. So with AAV gene therapy, you are delivering a correct version of that gene. It's expected to last for a long time. We don't know the time frame. Maybe it's 10 years, maybe it's 20 years. We'll need to sort of figure that out. In the ASO RNAi, you're not actually modifying any genes. You're just trying to, change the level of those. So that's not a permanent change. And then in CRISPR-Cas9, you are making a sort of a permanent change in the DNA of the cells where it's been delivered to. So ideally, if we have a specific delivery to motor neurons or Schwann cells, those cells would have permanent genetic changes. But it really depends on the mode of technology that you're using. So if you want to...
No, that's accurate.
And we have another question. Can this technology be directed towards mitochondrial neuropathy where the causative mutant gene is nuclear?
So that's a really good question. So the most common kind of mitochondrial-associated neuropathy is CMT2A, which is caused by mutations in the mitofusin 2 gene. One of the early efforts that we started in AAV gene therapy was actually on CMT2A. That effort sort of was stalled based on sort of investment of the company that was doing that. Zach's group actually has been working on applying CRISPR-Cas9 to CMT2A. Right now, I'm not aware of ASOR and AI strategy for that. But I would say CMT2A, being a relatively common form among the CMTs, has certainly attracted attention. And, I'm happy to tell you more about the different therapeutic options if you want to write to me about that.
Thank you ever so much, John. We're at time. So we're going to move to the final session of the day,